Alexander Ivanov from Northeastern and the Burnett Institute is going to tell us about low-flow, ultra-high performance chromatography that he is developing, which of course is one of the important technologies that enables to increase the sensitivity of mass spec analysis. As usually, he has the longest title of all presenters. I think that's quite clear. And I'm going to ask you to try to stay on time.
Thank you, Nikolai. Thank you, Bogdan, for inviting me here. It is a pleasure to attend the second meeting here at Northeastern. And I see that Bogdan and Nikolai are making a tradition out of this meeting, scheduling it right after SMS. SMS is a brutal, huge meeting, up to 7,000 attendees. I see many people came directly from this meeting. They look tired. So just a suggestion, next time, next year, if you continue keeping this tradition, it might be a good idea to organize the meeting in a slightly different venue, like peaceful environment, very relaxing, maybe a botanical garden, maybe a Japanese garden, maybe a spa. All right. So I'll be talking about ultra-low flow separations coupled to mass spectrometry. And we've been developing these technologies quite a bit. Last year, I showed some data, and I focused the talk on the sample preparation side of things. Today, I'll be talking more about ultra-low flow separations and coupling to mass spectrometry. So, and I'll start off with some of the results that we published a few years ago, and then I will show the data mostly generated by these 5 talented individuals from my lab, and I'm proud of their recent accomplishments and progress: Michal, Kendall, Tony, Annalise, and Somak. And I'll be talking about a limited sample analysis and our first steps towards characterization of post-translational modifications to some extent, at least we are starting, and how we improve the sensitivity of our proteomic and PTM profiling there. So, since we are back to the world of mass spectrometry-based proteomics, I wanted to start off with listing all possible fields where we have to be to bring our experiments to perfection. Sample selection, sample preparation, separation, mass spectrometry, data analysis, validation, and we have to make everything working well in order not to fail in the overall experiment, especially if we are talking about limited samples and single-cell analysis. This is very important. So, as Nikolai mentioned, it requires interdisciplinary, multidisciplinary teams, and so we cannot fail on any of these stages. So to briefly list fields where we need a limited sample, need to analyze limited samples. You can read here neonatal, microbiopsy, liquid biopsies, and so on. And we are at the single-cell analysis conference. I will not show you any data on single-cell analysis. We are working towards that. But we did our best over the years compiling the list. It's not incomplete, but at least different fields and different applications where limited sample analysis and high sensitivity proteomics is important. So also to emphasize the same point that Alex Kansas made before, other groups, and we showed before that slowing down, decreasing the flow rate of separation coupled to mass spectrometry increases the signal. Here in CE-based experiments, we did it in LC not long ago. And why? And Alex showed with 1 micron opening of the electrospray emitter. So we are trying to increase the ionization efficiency, ion transfer efficiency. We are trying to decrease ion suppression, and overall, it results in the increased ionization, increased sensitivity of our omic type of experiments. So also that was taken from the from a deck of slides prepared long ago. We are not using some of these parts, but I wanted to make a point that in order to run our ultra-low flow separation-based proteomic experiments, we are using pretty much standard approaches, even though here I'm showing a pico-clear union, which is not on the market anymore. You cannot buy it. But there are better new unions and crosses and T's that you can buy. Pretty much we are using a normal LC equipment, plumbed pretty much in a normal way. And we are developing either monolithic columns or plot columns. Plot stands for porous layer open tubular column. So you see the column is hollow, and a polymeric layer, porous polymeric layer, is attached to the fused silica capillary, or a porous monolithic column, as shown here. And now we continue developing both open tubular and monolithic columns, which I'll be talking about here, and I'll also show some result on capillary electrophoresis separation. A couple of points that we are making these columns in-house in our lab, and typically it is quite easy to polymerize the column and learn it rather quickly, plot column or monolithic column, operating it at 5 or 20 or 10 nanoliters per minute. It's more or less an art still. And we are trying to make it more robust. Even though we are using normal conventional equipment, we are trying to make it to optimize our methods the way that any lab can use it. And it takes time. We are not there yet. So it's and another introductory slide. Actually, here it's maybe already outdated. Plot columns. We are using now from 2 micron, 5 micron, 10 micron inner diameter tubes with attached stationary phase to the tube wall. And flow rate is actually now down to even 2 nanoliters per minute, up to 10 or 20 nanoliters per minute, which is not very conventional for typical non-LC experiments. But that's where we are trying to be. And we are trying to optimize the structure of the column to make them highly efficient, developing high efficiency of separation, high peak capacity, and that ultimately, together with ultra-low flow rate, decreased ion separation, we expect, should result in high sensitivity profiling. So, and we are using very small volumes of injections, which is potentially compatible or ideal for single cell analysis, down to picoliter range, nanoliter range injections. So where we were a few years ago, and I show it again as a starter, we compared our plot columns to an ultra-low flow mode of operation of columns with conventional columns. And I'll show more recent examples later. So that's where we started probably like 5 years ago or so, getting better numbers of identified proteins at that time using Q-Exactive and LTQ Orbitrap for a 10-fold less sample than we were loading on the conventional non-LC column operated in a more or less conventional mode. And that's just an ion density map showing multiple streaks, like thousands of them corresponding to peptides eluting out of the column and detected by mass spectrometer, and an M/Z mass to charge ratio on the other side. So at that time, and we published that a few years ago, we were able to get down to low attomolar levels, most likely even now reproducing the same setup, the same configuration, and using newer mass spectrometers will be also somewhere in the octomolar ranges of concentrations. We got to low attimoles, both in mass-MS and extracted ion chromatograms, which is MS1, stage 1 data acquisition mode. So, and to bring a reference to the plot that Nikolai showed and distribution of copies per cell, RNA versus proteins. So if you are talking about low zeptomoles, that corresponds to low thousands of ions detected there, and potentially with improved sensitivity of detection, we should be at low hundreds, and that makes the same point that Alex brought across and Nikolai brought across, that we should be capable of profiling single cells even without carrier channels. With carrier channels, it will be even better, but that's in principle it should be possible. So, and at that time also we played with injecting sample aliquots corresponding to 500 cells, and I show numbers again from 3 years ago or so, 4 years ago. And numbers of proteins identified here with tight correlations. And now we are trying to improve it, to improve the technology for column development and for LC. At that time, in each experiment, we were processing 2,000 cells and injected aliquots. Here, it was the same thing, 2,000 cells and injected aliquots corresponding to 50 cells, 100 cells, 500 cells, and so on. And then we coupled it to a more sophisticated way to prepare samples, to fish out samples from total blood. First, we whole blood. First, we spiked in cultured cells into blood, and then with target antibodies, we labeled the cells with magnetic beads, functionalized with antibodies, and fished them out and processed. And here, again, we started with 2,000 cells and injected aliquots corresponding to the numbers of cells shown here, 120, 200, and so on. And the number of proteins groups are shown here. The same approach was used to other endogenous cells, endothelial progenitor cells, and hematopoietic stem cells. Pretty much similar profile in depth, similar approach, and we were able to play with just samples derived from several donors and compare them quantitatively. Then with Jeff, it was a brief pilot experiment. Jeff Ager from Northeastern. We took a section from a mouse brain slice, carefully calculated cells, counted cells per that section. Again, sliced it into smaller section, and each section contained approximately 100 cells, according to the estimates and cell counts, plus minus 3 cells, I've been told. And then we processed the cells at that time using focused syndication and covariates, and injected an aliquot from a 100 cell containing tissue section, injected an aliquot corresponding to a single cell. And again, it's not a single cell experiment, but we get to the numbers close to 2,000 per one experiment. So now we are moving forward, and more recently, Michal and Kendall. Michal is on the left side here. He's the person moving forward the project on the LC side, ultra-low flow LC side. Monolithic columns, plot columns, and more conventional bead-packed columns. We continued optimizing recipes for polymerization of these columns, and we also compared performance of those columns, which I'll be showing. And Kendall was pushing forward capillary electrophoresis-based platform for limited sample analysis. And I'll share some results there as well. So back to the same principle of the decreased flow rate. When we are decreasing the flow rate using the same column, in this case, monolithic column 20 micron ID, from 80 nanoliters per minute down to 16 approximately nanoliters per minute, we see an increase in signal due to the reasons I described before and Alex pointed in his talk previously. So basically, we see what we expected to see. We keep checking ourselves, and here are the conditions. If we extract specific peptides, specific peaks, we see how the signal changes for them according to the same general trend observed in this slide. Then, of course, it can be translated to protein numbers, peptide numbers, PSMs, MSMS, spectra counts detected in each experiment. And we are using here a standard. We are not using single cells yet, but that's a standard that anyone can buy from Thermopeers. And it's a HeLa standard. We are injecting each time an aliquot corresponding to 6.7 nanograms, which is what, 30 to 50 to 60 cells on average. And if we are looking at these three flow rates and the same column, you see how the number of identifications, number of peptides identified, proteins identified, PSMs, and so on, increases when we decrease the flow rate. So we got here to approximately, what, 3,300 proteins, starting with 6.7 nanogram of HeLa. All right, it works. Then we are working on the surface chemistry. So we are trying to increase the hydrophobicity of the column to make sure that we are retaining even hydrophilic peptides on the head of the column. We are focusing them. They are not smeared through the column. And that's how it works. We first started with polystyrene divinyl benzene columns. Then we copolymerize C10 or C18 functionalities. It's still PSDVB based, but with addition of alkyl groups. So, and the signal intensity is more or less the same. The retention patterns are somewhat different. We see that when we inject peptides on the same HeLa digest on the C18, we see better retention. We see that more hydrophilic peptides are being captured, trapped on the head of the column. And actually, it's not only related to the hydrophobicity of the surface. We also see some morphological changes of our monolithic structures. So something changes there when we play with the recipe. So even though the compositions are very similar, nevertheless, the structure is changing. And how then it can be reflected on the identification results. In this case, I show just PSDVB with C10 PSDVB and copolymerized with C18. So, and I show here the loading step on the left, loading step here, and the numbers of peptides detected, loading and then gradient elution. And you see that on the left side, when we work with pure PSDVB columns, we have many hydrophilic peptides not being focused, detected during the loading. It means in more realistic experiments, they'll be lost if you have a trapping column or so, or they will be smeared. If you are talking about limited samples, we are getting worse chances to detect them. So, and here everything is pretty much shifted to the right. All the peptides are captured and well eluted, most of them, eluted during the gradient. And then the overall identification results reflect the same trend. So if we use higher hydrophobicity of the surface here, we are getting more proteins, more peptides, more spectra, and so on. And again, we are using the HeLa digest 1 nanogram in this case, and 1 nanogram of HeLa corresponds to either 10 or 5 or so, somewhere in this range, cells, I mean. And everything is shown at FDR below 1%. So we are climbing close to, what, 1900 probably proteins using 1 nanogram of HeLa and 1 hour gradient only. So we are not spreading here. We are just comparing the conditions, comparing the columns. Then, if we do the same thing for the plot column, porous layer open tubular column, the same trend stands, right? We are starting with 36 nanoliters per minute. We go down to 10, and in this case, we are using 10 micron ID column. Later on, I'll show smaller columns with smaller lumen. So the same trend where we see the increase of the signal when we decrease the flow rate is observed. And for this is base ion chromatograms, and for extracted ion chromatograms, it's pretty much the same. And now, if we look at 1 nanogram of HeLa and compare these 3 flow rates, we see an increase in the identification rate when we inject 1 nanogram onto the column operated at a lower flow rate of 10 nanoliters per minute. We are climbing up to 2,500 proteins identified in the in 1 hour long run, which is already a step forward in comparison to where we were a few years ago. And an example of a comparison of 3 types of columns operated at their more or less optimal flow rate or linear velocity rate. Base bead-packed columns, we are using Dr. Maish beads that John Neville is selling right there. He's not paying me anything, so he's sitting. And so we packed the column in-house. I think we got good at it. Monolithic columns, 20 micron ID. Plot columns, 10 micron ID. And you see how the signal intensity changes. And then, of course, it can be reflected in the identification results in the number of peptides and spectra we can generate, number of protein IDs that we can get. So again, 1 nanogram of HeLa, and you see how we can compare plot columns, monolithic columns, and bead-packed columns, 2,500 and going down to about 2.5 fold. Then, more recently, we switched to a 5 micron ID column, 5 micron opening of the lumen on the plot column. And here, we are injecting 0.14 nanograms of HeLa digest. And we are getting about 1,600, 1,700 proteins out of this injection. And the rest of the bars, they show similar injections using either plot column 10 micron ID, monolithic columns 20 micron, or 75 micron ID bead-packed columns. And 1 nanogram loaded into those, and 0.14 loaded onto the other one. Yes? In this case, it's not practical yet. We are trying to make it practical. And we are working on this, and it's still a proof of concept. So there are 2 ways. Of course, onto those columns, monolithic columns, and bead-packed columns, we are injecting directly. We load our sample directly. With the plot column, where we have higher hydrophobicity, we either inject directly, and then we still are not getting optimal results because we have a spreading of the sample along the column. Or here, in this specific show-off case, we are injecting with split. We are splitting the flow, like split injection. Not practical, but we are actively working on making that happening rather soon, injecting directly on the column or with the trap. We are trying to yes, come up with something where we can do it without wasting the sample. Here, we show that yes, we split, we get the numbers, it works in principle, the flow rate helps, the efficiency is there, not practical. And now, that's how we were moving from 50 micron to 20 micron ID, 10, 5 micron ID. And I hope that I convinced you that the next generation will be no lumen, 0 flow, 0 carryover, very good reproducibility. Yeah. What? Yeah, so that's our next generation. So maybe next year we'll be talking about this. So now on the CE side of things, capillary electrophoresis. Here we are not diluting the eluent coming out of the column. We are not diluting analytes. We are using sheathless electrospray interface, which is developed by the Maini group and commercialized by Sykes. Yes. So and and we are trying to get into PTMs here, and we've tried also limited samples, and I'll show you better other data later. So it works. There is separation. We are now able to spread the separation to 60 minutes to 80 minutes, which was a challenge before. That's how peptides elute from the migrate on the CE capillary. You see those pools, migration time, M over Z, and interestingly, how they cluster based by their net charge. And also interestingly, how phosphorylated peptides are getting separated without any enrichment. They are getting enriched based on their net charges and differences in charges from the rest of the peptides. They're moved to the right here, and we are identifying significantly. So first of all, we are getting a chunk of peptides that we don't see by LC, and we are getting many phosphopeptides we don't see by LC at all. So, and then we are also play with HeLa. We started playing with cancer cells, taking limited cells, and basically the same story is seen there. So, and here we treat cells to change their activation state and differentiation state with cytokines. With glycans, our first steps. Glycans are important. They're relevant, related to different pathologies. They can be up and down. So we are trying to shave them off the protein and analyze them by CZE-MS. So we are using this box, and again, John is right there, which is A-Bird. It's pretty much an activated carbon filter where we purify air and we introduce it into the region of a mass spectrometer. We use this one, and we use so-called dopant enriched gas, nitrogen gas. So we just bubble nitrogen through solvents and also introduce it into the electrospray region. So what we see with glycans by DP, I show degree of polymerization. It's like glycans. So signal in blue in conventional mode, A-Bird, and then gas. And smaller oligosaccharides, we see a trend in the increased signal. Bigger ones, a more pronounced gain in the sensitivity. And we are using up to 20, up to 40 folds increase. Then we optimize MS parameters. I'm not going into details, but they help us to get rid of adducts. So we first start with this distribution. Our signal is spread among many channels, and we are getting down to these, where we shook off all the adducts, and the signal is clean and nice, and it gives us a boost in the sensitivity, like 70-fold or so. And that's how the extracted ion electrophoregrams would look, conventional technique, and then optimized, smaller glycans and bigger glycans. And that gets to like 70-80-fold difference. Then we apply it to more realistic samples, IgG isolated from blood serum, and the same trend, increase in signal, and then we did the same with EVs, or here's another illustration. Ion density map, conventional signal, pretty much boring landscape, and substantial increase in the signal there. And then when we apply it to cancer cells, in this case, we inject an aliquot of 100 cells, similar as we started with HeLa, and we are getting over 150 IDs of glycans. And then we can quantitate them. With that, I would like to conclude. Yes, it still requires some work, but we are hopeful that miniaturized ultra-low flow separations will be very effective for limited sample analysis, single cell analysis potentially. We need to optimize. We showed good progress and nice identification results. New technologies are still needed, and for that, we need to join our forces and collaborate. And I think this is a nice platform, nice meeting to meet, discuss, and move forward together. Thank you for your attention. One most important slide too. I would like to thank my group members, collaborators, Barry, of course, and the collaborators from companies, Storma, and others, BU, funding, and of course, the group, the lab. Thank you.
David has a question for Alex.
Thank you. Excellent talk. With respect to your columns, the plot columns and your monolithic columns, have you experimented at all with mixed mode, where you might, for example, have the head of the column consisting of the monolithic column followed by a plot column to help with loading and then peak focusing?
Yes.
Cool.
What
I'm
kind
not
of
showing anything yet. Yeah.
what kind of pump are you using for maintain those flow rates?
This is a regular Dionix pump. We are, of course, talking to the same guys. Probably you are talking ProLab and all others. Here we just split the flow. We start with something like 200 nanoliters per minute or so, split it, and use, or maybe less, maybe 70, maybe, it depends. But we are not wasting too much solvent. We are using regular pumps. We are not under, so back pressure is very tolerable. It's very low for any of those. And actually, the length, if previously we worked with 10 meters, 8 meter long column, the latest results on a 5 micron ID plot was like 24 centimeter long.
But there's no pump off that could actually support flow rates directly below 50 nanoliters.
There are.
The zirconium pump. And they actually have computer automation. We can.
Just have a question. A very inspiring presentation. Thank you. Have you ever considered electrochromatography? That may also
considered what?
Electrochromatography. So when you're introducing the charge
yes, and we tried a little bit. It's like we tried, but with these sprayers, not that straightforward. So we tried. We still consider, we are not actively working on that, but I think it's, and previously, long ago with Barry and Csaba Horvath, actually, I published a paper on CEC.
That in principle can help you to introduce minute volumes of samples.
Possible.
Right?
Yeah. Yes.
Thank you.
Thank you.
Thank you, Alexander.
Thank you.
I'm still waiting to use your columns, as you know.
Great. Yes, I know. I know you are there. I know where.
The separation.
Where to find you.
The separation is one of the most critical aspects in our work. It's absolutely essential for high quality data, having good separation.